Well, hi everyone, and welcome to today's webinar on the basics of environmental monitoring programs. We're excited to have you here with us. My name is Jessica, and I'm the Director of Marketing and Sales here at Amtech. I'll be moderating today's webinar. First off, we encourage everyone to participate by asking questions throughout the presentation. You can submit questions by clicking on Q&A at the bottom of your screen, and we'll save some time at the end to go over all the questions that came through. We'll also be recording today's webinar, which will be made available on our website. I'll send out an email a little bit later to let you know when that's going to be available. To start off, for those of you who don't know, Amtech is an accredited laboratory in the San Francisco Bay Area, specializing in food safety testing, environmental monitoring, and special research projects such as shelf life, challenge, validation, and investigation studies. Today's webinar will be led by Morgan Young. Morgan is a microbiologist with several years' experience in food safety and pathogen research and industry experience creating and running successful environmental monitoring programs. We're also joined today by Dr. Florence Wu, who has extensive experience in pathogen detection, microbial contamination control, and environmental monitoring sampling strategies. And with that, let's get started. Morgan?
Thank you, Jess, and thank you everyone for attending our webinar today. Today's topic is about the basics of environmental monitoring plans. Food companies are required to develop and carry out environmental monitoring plans, but how do you develop your EMP? What are the necessary steps and essential elements? Today, we'll try to answer these questions and discuss how to avoid pitfalls, increase efficiency, and take meaningful samples. These are the main topics of today's webinar. We'll focus on two aspects of an EMP. One, the key elements of a sampling plan, and two, how to maximize efficiency of an EMP. It is important to note that this webinar is not intended to discuss all of the basic elements of an EMP, but rather to focus on the key elements that will help you to get started with your EMP.
Thank you, Morgan. And welcome again, everyone. And thank you for spending this hour with us. I hope is useful to you. When you are tasked with developing an EMP, the first question you would ask yourself is why? Why do we need to do environmental monitoring? What purpose does it serve? There are mainly three reasons: the reality of life, the needs to doing business, and a self-serving purpose. The reality of life is that we must be compliant with regulations. For example, FDA has requirements for preventive control and expectations for food safety. The need of doing business is that we must meet customers' specifications and audit requirements. However, I believe that a company's real purpose of doing environmental monitoring should be self-serving. A good environmental monitoring plan can help us to validate our environmental control and sanitation practice, validate food safety plan, and protect our brand name. Nowadays, bad news can reach millions of people in a few minutes. To produce wholesome foods that provide nutrition to people and do not cause illness is fundamentally important to a food company's business. Regulations and FDA expectations can help us to develop a proper EMP by serving as a framework of standards that including identify all the risks relevant to the food you manufacture, process, pack, or hold. Determine which risks need to be controlled to prevent foodborne illness, again, relevant to your food. Understand how to control the risk, whether in plant or in supply chain, and make sure that those risks are being controlled. And then finally, have a way to track that the system are working properly every day. If the main purpose of an environmental monitoring plan is to protect your food, the public health, and your brand name, and then the goal of environmental monitoring is to prevent cross-contamination. Let's look at the facility contamination risk. Many factors can contribute to facility contamination, including ingredients, machinery, people who work there, the environment itself, and your process, and then the history of the facility and the construction. So it is not only necessary, but very important to have an environmental monitoring program and perform regular sampling and testing so you are aware of the risk. The correct way to develop an EMP is that you and your team go through the process together. Firstly, learn and understand the government or trade association guidelines so you know the scope and what are the relevant factors. Secondly, perform a risk assessment to understand the risk of contamination specific to your ingredients, your process, your product, and your production environment. This requires intimate understanding and knowledge of your business that no one else has, and probably no one else cares as much as you do. Thirdly, it is advised that you collect the baseline data to demonstrate the microbial burden in your production facility and help you with risk assessment. Instead of speculating, you can just perform some sampling, send the samples to the lab, obtain some background or baseline data. It is the easiest thing to do and the low-cost effort to understand the risk of your environment. Hopefully, your EMP is developed by following the guidelines, assessing the contamination risk, and understanding the result of the baseline testing. A well-developed EMP should meet your overall food safety objectives. So what an EMP monitoring plan EMP should look like, and what are the major components? A complete EMP should have at least these components. The first is risk assessment. It's based on testing, sampling, and you have your reasoning for your sampling plan. A sampling plan that includes when, where, how often to sample, and what to test. And then you should have acceptance criteria that define what are acceptable, what are considered out of specification, whether it's a pathogen result or indicator enumeration. And then you need to have a corrective action plan to tell you what will you do if you have out of a specification testing result or pathogen positive result. And finally, there should be a review and verification process that should be performed periodically to make sure your EMP is up to date. We will look at these components in more details. The zoning concept is widely accepted as a part of a risk assessment. In this concept, area designates where the product is most vulnerable to microbial cross-contamination so that a proper level of monitoring and hygiene can be assigned. Each area is then divided into zones that refer to the location of swabbing points based upon proximity or closeness to an impact on product contact surfaces. Sites are the actual environmental sampling location with a zone that must be identified as swabbed. So this is a kind of a hierarchy of how you're naming your location and develop your sampling plan. Here is a diagram of a classification of zones that serves as example. Zone 1 is mainly product contact surface that can including slicers, peelers, fillers, of course, depends on your process, screens, conveyor belts, even employees' hand knives that, in direct contact with food. And zone 2 are non-product contact surface, but it's really close to the product contact surface. That can including exterior, under, or framework of equipment, refrigeration unit, equipment housing, and switches. Zone 3 is the other areas within the finished product room. That can including air return covers, hand trucks, forklifts, drains, wells. And zone 4 is the area that outside of ready-to-eat room. That can include locker rooms, cafeteria, hallway, loading dock, and maintenance areas. If the zones are classified based on risk level, then they should not be treated as equal in your EMP. So in general, we should sample more frequently, take more samples, and test more often in zone 1, zone 2, and less in zone 3 and zone 4. Performance standards are also higher in zone 1 than in other areas. Here is an example of a sampling plan according to the zoning concept. It is basically for small food manufacturer. So in zone 1, you can test ATP indicators, pathogen. If you have allergen control program, that might also include allergen testing. And the frequency can be daily, weekly, or combination of both. And the number of samples will need to depend on line and process, and the minimum 5 samples per production line. For zone 2, that is the area close to food contact surface. You can test ATP, or more commonly, people test indicators and pathogens, and it can be less frequent than zone 1. And the number of samples depends on the scope of manufacture. And zone 3 is further away from the process or open product area. And typically people test a pathogen on weekly or more frequent basis. Again, it's based on your production. And zone 4, you sample less frequently and really focus on pathogen testing.
Thank you, Florence. You did a really great job at explaining why we should have an EMP. And you talked a little bit on how to carry out an environmental monitoring program. But I wanted to talk a little bit more about exactly what should go into your EMP. Here are some requirements of an environmental monitoring plan. It must be scientifically valid. And then it also needs to identify those microorganisms, or if you choose just one microorganism that you're going to test for. You also need to identify specific locations from which samples will be collected and the number of sites that are to be tested during routine environmental monitoring program. The number and location of your sampling sites must be adequate to determine whether preventive controls are effective in your facility. You also need to identify the timing and frequency for collecting and testing your samples. And again, it must be adequate to determine whether preventive controls are effective. You also need to identify the tests conducted, including the analytical methods that are being used at your lab that you are using. And then you need to identify the laboratory where these tests are being conducted. And then you also need to remember to include a corrective action plan and procedure. When taking all of those requirements of a sound EMP, the fully implemented program should provide a baseline for microbiological assessment of a plant's environment. You also need to identify potential sources of microbial contamination, and possible vectors that may harbor or spread that contamination. You need to verify the effectiveness of your sanitation program. You also need to verify the effectiveness of procedures that are being used to segregate and control traffic. And this is traffic of personnel and equipment in your facility. Your program will also provide data to correct problem areas before they pose a risk to the product. It's very important that it is before that poses a risk to your product. And then lastly, it'll also provide trending data to modify and update programs in response to results and observations. So what else will we need to make an effective EMP? We'll need a few tools that will help us to obtain data and analyze it. These tools include visual inspection, ATP bioluminescence, microbiological testing, and other technologies such as DNA sequencing. Now, let's get into how we use our tools to meet the requirements we discussed earlier. One of the requirements was to identify harborage sites in your facility. We can do this through visual inspection. With this, you're going to inspect the processing area. You want to really examine for structural damage in your facility. You also want to watch the product handling practices in your facility. And so during processing, you're going to really watch those handling practices from your workers in your facility. You also want to watch the traffic flow of people and product. You can also watch your sanitation crew in process and post-processing sanitation. It's important to do both. And then you also really want to make sure that you are examining that product flow to really assess that contamination risk. It can happen at any point in your process flow. And then you also want to ask assistance from your line operators. They are the ones who know your facility the best. They know exactly all of those, like we said, that product flow. They know how the machinery works. So definitely utilize them. They are a huge, huge help. Another tool that we can use in an EMP is ATP. So what is ATP and what does it show us? ATP is adenosine triphosphate, aka the energy molecule in cells. ATP is present in all organic matter. And so we read ATP in relative light units. So following this process, more ATP means more light is processed through this luminometer that I have here. Luminometers, like this one pictured, are used to quantify the amount of light produced from a swab sample that was taken. This allows for acceptable limits to be set for your process, and it also enables data trending over a period of time. This is a great tool to validate your sanitation program. Now let's get into microbiological testing used in your EMP. When it comes to sampling in your facility, the big questions are always when, where, and how much. And we are going to discuss those right now. During pre-op, in general, this data should be easier to interpret. You're less likely to yield positive results. And when sampling pre-operationally after sanitation, it will identify sanitation weaknesses in your sanitation program. During operation, this is when you're more likely to yield positive results. Data from during operation will provide information on the spread of target pathogens during your processing. It's important to note that positive sample sites may not be the site where the pathogen survives. And then you will also require pre-op follow-up sampling to identify the pathogen source or niche. Lastly, post-sanitation, this is used to evaluate sanitation effectiveness. And something really important to note is that the FDA does recommend to test 3 to 4 hours within after production starts. So if you're doing during operation testing, definitely wait that 3 to 4 hours after your production has started. So how to take environmental samples is probably the easiest question to answer. You can definitely check out our website and also our YouTube channel to find several videos on how to collect samples, different types of samples, swab, water collection, air plates. Just feel free to take a look at our website, our YouTube, or you can always reach out to us and we can send those to you. Sorry about that. Okay. All right, so now let's talk about the actual testing of your samples. So where exactly should we test for pathogens, and when is it appropriate to test for pathogens in each location? Starting with zone 1, which, as a reminder, that's our food contact surfaces. It's recommended by the FDA that we do test zone 1 for pathogens routinely. We should also test zone 1 pathogen for pathogens for investigational purposes and as part of our root cause analysis if we do happen to find a presumptive positive. A few other important times to test for zone 1 for pathogens are whenever we have a new line, whenever construction or other major events are happening in our facility, whenever, of course, we find a positive for a pathogen in an actual food sample, and then whenever positives from pathogens in zone 2, 3, or 4, back to what I was mentioning with the investigational purposes. And then whenever we find troubling trends from our other aspects of our environmental monitoring program. So that's ATPs are really high. If you get any very high indicators, that would be a time to do more zone 1 testing for pathogens. Now we'll touch on when to test our zones 2 through 4. As a reminder, again, from what Florence mentioned, these zones are non-food contact surfaces. Zone 2s are those non-food contact surfaces that are directly adjacent to zone 1s. Zone 3s, those are our other non-food contact surfaces in our food processing area. And then, of course, our zone 4s are those areas outside of our food processing area. These should be tested routinely. And the pathogens that we should focus on are Salmonella and Listeria monocytogenes. We possibly might want to test for E. coli 0157 in some special circumstances, like if your facility kind of promotes the growth of 0157, or if you're dealing with a food product that is very prone to that contamination, that's when you would want to test for 0157. And then, of course, investigationally and for root cause analysis, that is another time that you definitely want to test those zones for your pathogens. Now I'd like to speak on the different foodborne pathogens that we were just talking about and the reasons for testing each. Salmonella, to start with, is responsible for 94% of low moisture food outbreaks. For example, in chocolate, infant formula, peanut butter, there's so many others, too many to count. It does survive in heat and dry conditions. So if you have those conditions in your facility, definitely consider swabbing and testing for Salmonella. Its harborage sites are where foods and dusts accumulate. So again, another great place for you to look in your facility, see where those harborage sites are. It can escape the dry cleaning process. So if you have a dry clean process in your facility, you definitely want to be testing for Salmonella. And it can also grow when those areas get wet, and that's when it just grows and grows. It might have a harborage point, but when you start adding water, that's when it can grow and then get into your food contact surfaces. Now on to Listeria species. Listeria species includes 6 species, and it is commonly found in soil. L. mono, that is the human pathogen that you always hear about. It really likes that cold and wet area. So if you have any of those in your facility, drains, chillers, that's where it's going to be hiding out. Its harborage sites include areas where food and water are present. So again, you want to look at those cracks and very hard to clean areas. So the special thing with Listeria that we really dislike it for is that it does grow that biofilm on processing equipment, and it makes it so difficult to clean. And then that's of course how it grows and gets bigger and goes into your product. Good sanitation and environmental control are critical to control this pathogen. And then just a little FYI for you guys, its prevalence in ready-to-eat foods is 0.17 to 4.7%. Next on to pathogenic E. coli. So that's that E. coli 0157 that I mentioned a little bit earlier. It does survive very well in the environment. For example, we hear about it a lot on produce. It is pH resistant. So again, in some of our fruit juices, those higher pH, or so should I say lower pH, it is resistant and can really survive in those. It has been isolated from food contact surfaces. So we find those in meat processing facilities, food contact surfaces in produce plants, things like that. And it can survive in low moisture foods. You hear about it a lot when you're talking about cookie dough now. You don't want to eat the cookie dough, not just because of eggs, but also because of the flour, because it can carry E. coli. So do keep that in mind if you're processing some of those lower moisture foods in your facility. So aside from pathogens, quality indicators are also a very valuable piece of your EMP. Zone 1 sites are typically tested for aerobic bacteria, coliforms, the generic E. coli species, Enterobacteriaceae, and or yeast and mold. Zone 1 sites should be swabbed pre-operationally before sanitation and prior to startup of the production line as a measure of cleaning effectiveness. When using a quantitative indicator such as an aerobic plate count, performance targets and acceptance limits should definitely be established. And I'll just touch back on our ATP tool that we talked about earlier. The effectiveness of general cleaning and sanitation can also be determined rapidly on site using ATP. A large number of sites can be evaluated very quickly using this. So either a 2-class pass-fail or 3-class acceptable, caution, fail sampling plan can be used for your ATP. Similarly, if our allergens are a concern for your facility, you're using nuts, you're using wheat, and then you have some some products that don't have those, and you want to mark, that they were processed in an allergen facility and you don't want to have that cross-contamination, you can use a rapid protein assay, and most of them take about 15 minutes to use. So I wanted to go in a little bit more detail about some of the quality indicators that we can test for, starting with aerobic plate count. APC is an indicator of bacterial populations in a sample. APC includes bacteria that are aerobic, mesophilic, and they're grown on standard agar. APC can be used to evaluate sanitation methods, adhere to GMPs, determine organoleptic acceptability, evaluate shelf life, identify spoilage, and then also evaluate conditions during processing, storage, and handling. Now on to Enterobacteriaceae. Enterobacteriaceae is a family of bacteria that are gram-negative, facultative anaerobes. Some of the notable pathogens that are in this family include the pathogenic E. coli, Salmonella, and Shigella. And many species from this family are commonly found in gut microbiota of mammals and in plant material. So if you are processing any sort of mammal or any sort of plant material, that is definitely a great time to test for Enterobacteriaceae. Enterobacteriaceae is tested as an indicator of pathogens such as Salmonella. So it can kind of help you to realize that there's a problem before you have a problem, kind of like what we mentioned earlier. Next, on to coliforms and E. coli, and this is the generic E. coli species, not pathogenic E. coli. Coliforms are a gram-negative, non-spore forming rod that ferment lactose to acid and gas within 48 hours at 35 degrees Celsius. Getting a little bit technical, just so you guys can kind of understand how we test for those. Fecal coliforms ferment lactose to acid within 48 hours at 44.5 degrees Celsius, so a bit higher temperature. E. coli, the generic species, is an indicator for fecal contamination, and some E. coli are pathogenic. For example, E. coli 0157, and then you also will hear a lot of people talking about the Shiga toxin producing E. coli or STEC. That's another group of E. coli that you can be on the lookout for because it does make some people sick. So now on to yeast and mold. It can be used in our testing plans to evaluate food spoilage and decomposition, determine shelf life, adhere to GMPs, and then also evaluate processing and handling conditions. And just wanted to mention, these are definitely an indicator of moisture. So if you're doing a low moisture food in your facility, this might be a great way to just go ahead and check that you're not getting added moisture into your low moisture product. So now, knowing all this information about the different types of quality indicators, you can choose which indicator or indicators that would be best to test for in your facility. As I mentioned previously, it is important to establish performance targets in your operation. These performance targets will help you to evaluate the effectiveness of your sanitation program. Different operations will have different targets. Baseline testing is super important in establishing your targets. This is just an example of a possible scenario for your operation, but it is important to do that baseline testing, like I mentioned, in your facility to determine the acceptable limit for your specific operation.
Thank you, Morgan. You have discussed the sampling techniques, when, where, and how often to sample and what to test. Now, I would like to discuss how to establish a baseline. This involved 3 steps: collect data, analysis, and set limits. To establish baseline data, you want to sample when the system is under control, not right after there was big construction or switch into new procedure or process. You want to do it when under normal production, when the system is running properly. And then you want to collect at least 20 samples. So 20 sample is kind of requirement for basic statistical analysis. And then for the samples collected, you want to do testing. The testing can be as recommended as by Morgan, that whether it's indicators or pathogens, but for pathogens, is when you don't have a number, when it's just presence absence, then statistical analysis could be difficult. So indicator testing is usually performed to establish baseline data. And then data analysis that including the testing result, and also use Excel, for example, for simple calculation. The simple measure you can do is the average or the mean or standard deviation. To set limits, there are different ways to set the limit, but there are 2 common ways, 2 common measures you can set. One is upper warning limit, which is the mean plus 2 standard deviation, and upper control limit, that is mean plus 3 standard deviation. So let's assume you have established baseline and use it to evaluate the performance of the environment. And then you keep sampling and testing. Over a period of time, you have accumulated a lot of data. What do you do with this data? The smart thing to do is to analyze the data for trends, get more insight for your environment, and identify any patterns. It would be great that you can use your past data to predict future impact of sanitation or production activity. There are many resources for tracking and trending EMP data. The use of Excel is a popular means for tracking data since almost everyone can use Excel to some extent and has Microsoft Office software. Excel can do a lot of data analysis and produce charts that are easy to understand. Many companies have partially automated software built into their sampling device. Some systems are highly automated, and both hardware and software fully integrated into their overall food safety management system. There are also standalone software products that are tailored for managing environmental monitoring program. Laboratory information management system, LIMS, can also be customized to track environmental testing data. If you work with a third-party lab, you can seek their help to generate trending report. Here is an example of a simple data trending use Excel. The chart on the left is the AEROBIC plate count and yeast mold count from exposure plate testing. The chart on the right is the monthly variation of out-of-specification result. If analyzed properly, the data trend may be associated with event or seasonal change or production volume. In this case, it definitely shows, the AEROBIC plate count, the yeast mold count has seasonal variation. This is in California, so in January, February, we'll probably see higher count in airborne microorganism because the rain promotes the growth of vegetation and promotes microorganism in the air. And on the right, the pathogen, this is related to the production activity, the switch over of products or switch of different ingredients. Here is an example of a standalone EMP management software developed by Fremont. That's a company that has a state-of-the-art EMP system that including electronic sampling valve and software that has automatic data trending. If you use that software, this can be done pretty easily.
Thanks, Florence. Yeah, from my own experience, entering data and doing the data trending manually is very, very time consuming. So yeah, really excited about Fremont's automatic sampling and data management system. That's really, really great. Unfortunately, though, there are parts of an EMP that just cannot be automated. And that's when we need to deal with contaminations or those out of spec results. When contamination in the facility is above the designated control limits, or when pathogens are found, investigational sampling will need to be started with a much higher frequency than that routine environmental sampling that is included in your plan. Initially, contact and indirect food contact areas, they will need to be emphasized to find the contaminated sites, and you really just need to deep clean them. Then the intensified sampling will spread to your adjacent zones. The line will be stopped. The equipment should be deconstructed for sampling and deep cleaning, and it is essential to include perimeter zones and non-process areas in the sampling, since these areas can often be vectors for contamination due to traffic within your facility. The chart showing high-risk sampling sites is useful as a guide for starting your sampling. Okay, so what exactly do we do if we have that awful pathogen positive? It's really important to remember to hold and test product from that production day or the days that that positive sample was taken. You want to really examine your equipment that tested positive in that surrounding area. You want to review and modify your HACCP or food safety plan. That's an important part of, reviewing and doing that corrective action. You want to intensify sampling and testing for a minimum of 3 consecutive days. That is another recommendation that comes from the FDA. It comes from a lot of us testing labs. You want to do that 3 consecutive days to really make sure that you have gotten rid of that problem. You want to test upstream and around that positive testing location. This is called vector analysis. You want to check maintenance records for major equipment and disassemble that equipment if necessary. You should definitely be really close with your maintenance team because they're going to help you a lot, just like your sanitation team. You're going to interview and observe sanitation. Again, kind of that cross-functional team working on your EMP to make sure that you're getting these problems solved. And then review and modify production, maintenance, and sanitation procedures as you see fit. So further actions to be taken in responding to a positive pathogen result are identifying potential causes. You need to do an extensive review of your traffic patterns, equipment layout, and adherence to personal hygiene procedures, and that will really help you to find those potential causes. If product or environmental, if you have a product or an environmental positive, you want to reprocess, divert, or recall your product. Stop production. That is a key point here. You want to stop production just to really evaluate everything that's going on. And then you can always consult a food safety expert. If food contact surfaces are negative for 3 consecutive days, product from those days can be released. A similar approach should be taken if a positive is found on a non-food contact surface. For those investigations, you'll want to concentrate on your zone 2s, conduct a root cause analysis. Of course, that's anytime you get that presumptive positive, and then determine if you need to disassemble equipment, which I've found in my experience is always helpful to just go ahead and disassemble equipment because there's always something hiding in there. When a positive is found, you need to do vector swabbing. And it is conducted in a starburst pattern, 360 degrees around that presumptive positive site that was sampled. If additional positives are found, the vector swabbing will be extended to determine the extent of contamination. So for example, if you get another positive 10 feet away, you should also vector around that other positive that was 10 feet away from your first one. Any item is a suspect that is in that immediate area of the processing line. Any open cabinets, open panels, wheels or legs on equipment moving in and out of that area is suspect. So definitely keep an eye on those moving carts and wheels that you have some of your other parts of your production coming in and out of that area. Vector swabbing is used so that we don't forget to look up and above the product or below the product, where those niches and harborage sites may have developed for those pathogens you're looking for. Any drip, condensate, or airflow over product is suspect, including compressed gases. So that is one thing to always remember. Look up, grab a flashlight if you need to look under your equipment as well. Just really go in on that investigation and look all around you. So what do recurring positive results mean? Detecting the organisms in several sampling locations during the same sampling period, it could indicate that there is something in your routine sanitation practice that is just not adequate. And it also means that there might be potential harborages. So again, you might have to get multiple teams in your facility in on this investigation. Maintenance, facilities, sanitation, they can all help you with really getting to the bottom of this. For example, there was a salmonella suspect that we have helped with that was found in a facility for over 10 years. So you really got to make sure that you're identifying if there is a resident strain and not just a transient strain. Speaking of those transient strains, those are strains that are just passing through. So if we go ahead and identify that transient strain, then we can eliminate it before it does become that resident strain. And of course, finding that presumptive positive, it will increase the risk of decrease the risk of cross-contamination from zone 2 to zone 1. So if you do find that, you want to really eliminate that risk. Sometimes corrective actions just simply do not work. In spite of every effort to monitor, sample, and correct environmental issues, including deep cleaning equipment and environments, production lines still test positive erratically for pathogens or pathogen indicators such as Listeria species. Such equipment should be segregated and removed until your solution is found. The facility should consider taking the equipment out of service if it cannot be cleaned properly. So just keep that in mind. We don't want that to happen, but hopefully with your EMP, it will catch it before it gets to that. So just to summarize a bit, there are two main takeaways from our talk today. A food processing environment is ideal for the survival of pathogens. Key pathogens to remember are your Listeria monocytogenes and Salmonella. Pathogens that persist in the processing environment have been linked to costly recalls and outbreaks. So if you catch it before, that's fantastic. And then finally, environmental monitoring programs are a critical component of food safety. All processing plants should have an EMP, and we are seeing that from a lot of auditors that are coming in to help with third-party audits at facilities. And then results need to be used for immediate corrective actions as well as long-term improvements. If you enjoy, oh, sorry, not done. If you did enjoy today's webinar, please join us for our Advanced EMPs webinar that Florence will be hosting. This webinar will do a deeper dive into using the data from your EMP to further understand your processing environment. And I know we kind of did a very short breeze over of what to do when you find those dreaded pathogens. And in this next webinar, we will also cover developing a sound corrective action plan to mitigate your out of spec results.
Thank you so much, Morgan and Florence. Sorry about that. We did have a few questions come in during the presentation. The first question is, should an EMP include both indicator and pathogen testing, or is just indicator sufficient?
Oh yeah, I would love to talk about that. I believe that you should include both. Those indicators will really help you to, like I said, focus on that sanitation validation, and that can also help you to find the pathogens before the pathogens are a problem. If you're noticing an increase in your indicators on your food contact surfaces, that can kind of help to show you that you might have a microbial problem in the rest of your facility. Definitely, I do want to say you want to do those indicators on the food contact surfaces, not really the rest of your facility, because you're going to have high microbial indicator counts in other parts of your facility. But if you're doing it on that food contact surface, it'll really make sure that you're keeping the most important part of your facility very, very clean. Florence, do you want to speak any more on that?
Yeah, as we talk about the zoning concept, so different zones, they pose different risk to contaminate the product. And also your sanitation had different emphasis, right? So how you evaluate effectiveness of sanitation, and how you're going to evaluate whether there is a niche or there is biofilm found somewhere, and those can be done by indicator testing. And then the pathogen, of course, we need to test the pathogen, especially in zone 2, 3, and 4. So we know they're not in the environment. So I think they're serving different purposes, and those purposes are important to EMP.
Okay, got a few more questions here, so let's try and get through those. The next one is, what is the baseline acceptable bioburden in the food processing environment?
I think that's the number. This is Florence. I often get asked by that number. There are some guidelines out there, but FDA does not have a standard per se. So you do want to develop based on your experience, your product. For example, for the contact surface, if your sanitation include both cleaning and disinfecting, then you want to really shoot really low number. And low number, it can be for example 100 CFU per 100 square centimeter. So that's the thing, the important thing to remember in the EMP. When you define the acceptance criteria, you need to define the area. You can, the area that you want to compare. So each time you sample the same size area, or you actually record area, so you can calculate the CFU per square centimeter, or per square inch, or per square foot. So have the standardized data, and you can compare from time to time from different sampling events, and then also you can establish the acceptance limit. I see food companies, they establish limit as like for ENTEROBACTERIACEE, for example. In some production, they don't allow any of those. So you want to establish lower number, maybe less than 10 CFU per 100 square centimeter, versus versus APC can be a little bit higher, because APC is a bigger group. So that number, that acceptance limit, should be relevant to your production, your sanitation practice, and also relevant to what you're actually testing. And of course, for pathogen, you want to be non-detect, right?
Mm-hmm.
And the next question is, what considerations result in classifying a microorganism as objectionable?
I'm sorry, can you repeat that, Jess?
Yeah, what considerations result in classifying a microorganism as objectionable? So why would you classify a microorganism as objectionable versus not?
Florence, do you wanna? Yeah, go ahead.
Yeah, that's a really broad question. So I hate to say depends, but it really depends. For example, if your products fall into the USP, category. And they actually have a list of objectionable microorganisms that including E. coli, Pseudomonas aeruginosa, Salmonella, Clostridium, even Candida, which is the yeast that cause disease, right? So they have their list of objectionable microorganisms. And then you have FDA is saying that Salmonella, Listeria mono have zero tolerance for those. So obviously they are objectionable. And then you have beef industry look into more of E. coli 0157 or rest of the stack. So basically, if you just define it, objectionable bacteria or fungi are the ones can cause health, health problem in human. But the actual species or actual group is relevant to the industry guideline and your product and of course regulation.
Thank you, Florence. The next question that we have, we are introducing a new line in our facility. What steps should I take to introduce it into our EMP? So how can we,
I think that's a great question. The nice thing about a new line is that you almost get to start from scratch. So I would definitely start off by doing some baseline monitoring. I personally would call this kind of a commissioning. So you're commissioning the new line into your facility. After sanitation of your new line and the new line equipment, definitely take indicator and pathogen swabs, swabs to be tested for those indicators and pathogens. It's a great time to get that initial baseline swabbing of the indicators that will help you to figure out what your acceptable pass limits are. And then also it would just make sure that you're not bringing any sort of unwanted bacteria into your facility as well.
Thank you, Morgan. So I think we're out of time for today. We do have a few more questions. So if you didn't get your question answered, we will reach out to you personally and answer those questions for you. So don't worry about that. But I want to thank you so much, Morgan and Florence, for your time today. We also encourage anyone who's interested in EMP assistance or testing to reach out so we can schedule a meeting to discuss your needs. We'll also send you guys an email so you can book a time to meet with Morgan if you need anything to discuss with her. Again, thank you so much for attending, and we look forward to seeing you at our next webinar in June. That's going to be the Shelf Life Studies Part 1, Microbial Spoilage. And again, we'll also reach out with information on the Advanced EMP webinar that's going to happen in October. Thank you guys so much, and hope you have a great rest of your day.